Hi everyone, my name is Anne, and in this video I'll be showing you some basic pre-processing steps to get you started with ERP analysis. So when we're pre-processing data, there are two main goals. The first goal is to reduce the noise in your data so that you can see the effects of interest more clearly. And the second is to get your data in a format that's appropriate for measurement and statistical analysis. So with respect to noise, noise is basically activity in your data that you're not interested in. So this can be like non-brain sources of activity, such as line noise interference, or it can also refer to brain activity that's not task relevant. Basically, it's not practical to get rid of all of the noise in your data without also getting rid of some of the signal. So try not to get too carried away with eliminating noise in your data. You just want to reduce it by getting rid of the larger and more obvious sources of noise. So in this video, I'll be talking about how to load your data. I'll be talking about referencing, filtering, dealing with bad channels, and artifacts like eye blinks, and how to extract epoch segments. And then I'll finish by showing you some plots of the data that we processed. Okay, so let's have a look at some data and I'll explain things as we go along. So the first thing you want to do is open EEGLAB by typing EEGLAB in the command window. And then you'll see a window that looks something like this. And the next thing to do is to load the data file. Yep, so today we'll be working with some sample data, which you can access from the link in the description. And today we're working with .trc files. And to import the data, you just go to File, Import Data, and we'll be using the File I/O interface. So each EEG system is different, and you may have to use a different function depending on which file that you have, but we'll be using this one. So just click here, say that you want to import a file, and then we navigate to the folder, click on the file and hit open. So we can just hit OK and use the default parameters. And it's going to ask you about the data set. We can just ignore these and just hit OK. Awesome. So once you've loaded your data, you can see that there's some information here about the file. So the data contains 60 channels. It has one epoch, which means that it's a single continuous recording and that it was sampled at around 1000 hertz. And down here, you can see that we don't know what the reference is yet or what the channel locations are. So once you've loaded your data, it's a good idea to inspect the data just to see if your event markers are there and whether there are any unused or mislabeled channels to deal with. And also, when you're learning, it's good practice to inspect your data after each step just to see how your data is transformed. So to plot the data, you go to Plot and click on Channel Data Scroll. And a window will pop up like this. Okay, so this is the raw EEG data. Each line reflects the activity of a different electrode, which is labeled here on the y-axis. And this is plotted over time in seconds. So here we've got 5 seconds worth of EEG data. So one thing you can notice is that all of these traces look quite similar. And this is because the EEG has not been properly referenced yet. You can also navigate on the plot by pressing the arrow keys. So if you press the right key, it'll go across. And you can notice that we have some event markers here, which tell you when something has happened during the experiment. So in this sample data, participants did an experiment where they were presented with odd and even numbers, and they were only required to respond if they heard an odd number by pressing a button, but not to do anything if they heard an even number. So even numbers are represented by 100, odd numbers by 101, and the response is marked by 111. So before we can proceed, we do have to exclude this final channel here, which is unused, and we have to relabel the EB channel, which stands for eye blink. We need to rename it to something that EEGLAB can recognize the position of. So to delete a channel, you need to go to Edit, select Data, and a window will pop up. You just need to click this button on the bottom row and select the channel that you want. Hit OK. And then you need to check this mark to say that we want to delete that channel. Once it's done, you hit OK. It will ask you about the data set again. We just ignore that and hit OK. And so what you can see now is that the number of channels has decreased from 60 to 59, indicating that the channel has been deleted. So the next thing we wanted to do was to rename that EB channel. And to do that, you go to Edit and you click on Channel Locations. And a window will pop up asking you about which coordinate file you want to use. So EEGLAB comes with some files that contains standard electrode locations. And so we want to select the one that says spherical with eye channels. And then you hit OK. Once this is done, you'll get another window, and on this window you will see that there is the electrode label, and that's accompanied by coordinates in 3D space, which tells EEGLAB where the electrode is. So what we want to do is we want to keep clicking until we find EB, which should be somewhere in the middle. Okay, yep, so this is EB, and you notice that there's no coordinates because EEGLAB doesn't recognize EB as a standard location. So we're going to change that to IO1, which stands for the infraorbital region of the left side. And once that's done, we can click Lookup Locations for it to find the coordinates. And again, we want to click the one that has spherical file with eye channels and hit OK. And now you can see that the coordinates have been populated. And so that's pretty much that. So you just hit OK. And now that that's done, you can see that the channel locations has been changed to yes. And just a final point, channel locations are important for if you want to plot scalp maps, and I believe you also need it for independent components analysis. So the next thing that we'll do is we will re-reference the data. So when we're working with EEG data, we're dealing with voltages, and there's one thing to know, and that is a voltage is a difference in electrical potential between two electrodes. So this means that voltage is a relative measure. So a voltage at one electrode is always relative to another one. So electrodes that you're interested in, sometimes they're called active electrodes, and the electrode that you're comparing to is called the reference. So due to the relative nature of voltages, the reference is generally placed on a site with less electrical activity. So sometimes you'll see that people will place the reference on the mastoid bone, which is the region behind the ear, or on the tip of the nose. Alternatively, people also use something that's called a common average reference. And basically, a common average reference is when you use the average of all other electrodes as the reference instead of a single electrode somewhere on the head. And this is just based on the assumption that the head is a electrically closed system. So if you have electrodes all around the head and you average them, then all the positives and negatives should cancel each other out, giving you a net zero, and that's what's being used as the reference. So I won't go into any more detail. I'll just show you how to compute an average reference. To do that, you click on Tools, you click on Re-reference. So we'll be using the average reference, which has already been computed. And you also have the option to exclude some I channels. So we'll click here and do that. And then you hit OK. So generally I channels are a bit noisy, and so that's why you might want to exclude them from the reference. Okay, so now that that's done, we can actually inspect the data again. And you can see that the EEG traces look different from before. And you can see that channels, there are two channels that are a bit noisy. So this is T3, and this one you can't really tell, but it's actually IO1, which is the iBlink channel. So the next thing you could do is find a way to deal with these bad channels. But before we deal with those channels, I like to filter the data. And this is because for many data types, the raw data isn't nicely positioned on the plot like it is here. So some of the lines won't even be on the plot. And so it would actually be very difficult for you to figure out which channel was bad. So that's why I filter the data before fixing the bad channels. Okay, so with respect to filtering, so filtering allows you to reduce noise by filtering out the lower and higher frequencies where the noise is present. So this can help with slow drifts that occur throughout the recording, or high frequency noise that might be caused by external interference. So here I'll show you how to do a basic 1 to 30 Hertz filter. So 1 to 30 Hertz filters are easy to work with, and they're generally fine for ERP analyses, but you can widen the range to suit your application. So to filter the data, you go to Tools, Filter the Data, and Basic FIR Filter. Then we just enter 1 as the lower edge, 30 as the higher edge. We can uncheck this and click OK. You can ignore that, hit OK. And so now the filtering is done. We can plot the data again. And so you can see here that the data is a lot smoother. And now what we'll do next is we'll reject some bad electrodes. So we said before that T3 was noisy. So T3 is not too bad, but we'll just interpolate it for the purposes of this tutorial. And if your electrode is noisy due to imperfect contact, this can increase the noise of other electrodes if you're using the common average reference. So that's why you would want to interpolate. Filtering can help with noise like it kind of did here, but it's not an effective way of dealing with bad channels. And so what you can do is you can interpolate the channel. So interpolating basically means to get rid of the data on that channel and replace it with data that's reconstructed from the surrounding electrodes. So an important thing to note here is that the interpolated electrode no longer contains unique information. So if you have a bad channel that's critical to your analysis, then you might want to consider excluding that participant instead of interpolating. So to interpolate, you go to Tools, and you go Interpolate Electrodes, select Data from Channels, and you click on the one that you want, and it'll come up here. You use the spherical method and just hit OK. And so one thing is that the eye channel was also a little bit noisy, but I don't like to interpolate eye channels just because electrodes on the edges of the scalp don't really interpolate as well. And also it's the only channel that is measuring the eye movement. And so if you interpolate that, you pretty much lose that channel. We can plot again to inspect the data. And you can see here that T3 is free of noise now. Yeah. And one other thing you can see now is that the data is quite clean, except for these peaks here, which reflect eye movement, eye blinks. And you can see them a lot better when you stack the waveforms. And so that's when the participant blinks or move their eyes. So we can reject these artifacts by performing what's called an independent components analysis, or ICA. And then after ICA is done, we can reject components that we think are associated with the eye blink. So ICA is a blind source separation technique, which basically identifies patterns in your data, which it calls components. And so because these components are maximally independent, it's possible to reconstruct the signal while excluding certain components. So this process can take a while depending on how much data you have and how fast your computer is. So it can take anywhere from a couple of minutes to about an hour per data set. So one way to reduce the time that it takes for ICA is to downsample the data. So for ERP analysis, we don't really need 1000 Hertz. So what we can do is downsample to 250, which means that you will have less data points. So rather than having a data point every millisecond, you have one data point every 4 milliseconds. But this is still more than enough. So to downsample your data, you click on Tools, change sampling rate, and you just type 250. Then you hit OK. And just to plot again. So you can see that the data doesn't look any different. Okay, so to do ICA, what you do is you click on Tools and Decompose by ICA. So we'll be using the Run ICA algorithm. And normally I use something called Amica, but that requires some extra installation and it's a bit out of the scope of this video. But for the purposes of this one, we'll just use Run ICA, which pretty much does the same thing. So just hit OK. And if you've interpolated a channel, you're going to get a window saying something about the rank being reduced. And yep, this is because when you interpolate a channel, the data at that channel is no longer unique. And it's just saying that EEGLAB has recognized that. And so the implication is that ICA returns components, as many components as you have unique channels. And so if you interpolate a channel, you have to decrease the rank, which will return one less component. And that's fine. So we'll just hit OK. And the ICA will run. And we just have to wait for the ICA to finish. So another thing you can do is to extract epoch segments before running ICA, which will help it run faster. And when you extract epoch segments, you're basically getting rid of the data in between trials. But we won't do that for this tutorial. Awesome. So now that ICA has finished, you can see that we have ICA weights now, and now we can reject components associated with the eye blink. So to reject eye blinks, you go to Tools, Inspect by Components, and it'll ask you how many components you want to plot. So we'll just plot the first 20. So components are ordered by the amount of variance that they explain. And because we're only interested in the larger sources of noise, such as iBlink artifacts, it's only necessary to consider the first 20 or so components. Because ocular artifacts are very large, they almost always show up in the first few. And you can tell which one is an eye blink based on its frontal distribution. And you can also tell by looking at its time course. So to inspect, you click on this. And so this is the scalp distribution. And over here is the time course of activity. So time goes across like this, and then it keeps going up. And red refers to the positive amplitude, and blue is the negative. And so what you can see here is that blinks are bursts of activity that are pretty randomly distributed, as you would think that an eye blink is. And so if we're happy that that's an eye blink, you just click here to flag it for rejection and hit OK. And so you can see there is another eye blink component, and it has similar activity, so we can flag that. And so I think what's happening here is that this component is flagging the first half of the experiment, whereas this one kind of captured the second half, which can sometimes happen. And there is also another blink component, another component that's here that's related to eye movement. And so what this one is capturing is actually horizontal eye movement when the participant moves their eyes side to side. And so that's marked by this kind of distribution here, where half is blue and half is red. And if you look at the time course, it's normally like a rectangle like this, and you will have alternating red and blue as the participant moves their eyes to the side and back again. So we will reject this one. And that's pretty much it. So once that's done, you hit OK. And so what you have to do after this is to go to Tools and Remove Components. So the process that we did before was just to flag the components to be removed, but this step actually removes it from the data. Once you do that, it'll give you the option to confirm, and you can click on Plot Single Trial to have a look at the data and how it's different. So the red line represents the corrected trace, and the blue line represents the original trace. And so if you stack them, you can see how the data looks when you reconstruct it without those three eye movement components. And you can see that it does a fairly good job of correcting for the eye blink. Once you're happy with that, you can close that and click accept. Hit OK. Cool. So now we've pretty much done most of our pre-processing. And the next step is just to split the data into epoch segments. So when we're working with event-related potentials, that means we're interested in brain activity that's related to specific events, like the presentation of a stimulus. And we want to see whether that activity differs across experimental conditions. And so by looking at ERPs, you can make interpretations about what the brain is doing or doing differently between conditions. And to do this, you consider the task that the participant is doing, the location of the effect in space and time, and maybe by examining the relationship between these potentials and behavioral performance metrics like reaction time and accuracy. So to extract epochs, you go to Tools and Extract Epochs. And then you click on this button to say what events you want to time lock to. And so we will be extracting events to both the standard and the target, so the even and odd numbers. And you click OK. And you can define what time window around the events you want to extract. So the default is -1 seconds to 2 seconds. So that's 1 second before to 2 seconds afterwards. And what we can do is we'll just do 200 milliseconds before to 1.5 seconds afterwards. And then you hit OK. And then you hit OK. And the next thing, it will ask you to do a baseline correction. And so normally you just baseline to the pre-stimulus interval, which is the period before the event that you are interested in. So you just hit OK. Cool. So one thing I would like to mention, which is, so one thing that I do that's a bit different from others is I like to extract the epoch from all trials and conditions at the same time. Other people can sometimes will extract like one condition at a time. And the reason why I do everything together is that it preserves the order of trials, which is important if you want to do trial level analyses. And then what I'll do is I'll merge the EEG data with behavioral data in my Excel spreadsheet, and I use that to keep track of my data. So yep, I'm extracting every single trial from the experiment, and you can see here that there are 240 epochs, and that corresponds to there being 240 trials in this block of the experiment. So if I compare that with my behavioral data, so this is the data here, and so you can see that for each row it tells you what type of trial it is, if there was a response, what the response time is, and you can see here that there are also 240 trials. So if there's 240 epochs in the EEG data and 240 trials rows in the Excel spreadsheet, you can assume that there's a one-to-one correspondence, and then you can use this information in the spreadsheet to split the data into different conditions. So what I'll show you now is how to import Excel data into MATLAB, and you can do that using this script here. So you can use the read table function, and all you have to do is say where the file is located, and you have to say what sheet that you want to extract from. And we're storing that data inside the EEG structure. And so once it's done, you just enter. And once that is imported, you can find that inside the structure where we saved it. And you can see that all that information is now here. Okay, so now that we have extracted epochs and imported the behavioral data, we can plot the data again to see what that looks like. And so what you can see is that rather than it being plotted over times in seconds, what you have is that it's plotting it by trials. So you have 1, 2, 3, 4, 5 trials, and you can see how it's time locked to the stimulus. So this is an even stimulus, and you can see here that time in milliseconds is 0. And we extracted 200 milliseconds before that and 1.5 seconds afterwards. And you can change the scale. Okay, so you can see here that there are still some artifacts that are left. But if you keep scrolling, you can see that there's not very many of them. And so you can flag those trials for rejection by using another technique. But I won't cover that in this video. So that's pretty much all of the pre-processing steps that you'll need to do. And so what I'll show you now are some waveforms and plots of the data so you can see what we've done to it. So I do have some code here that will plot the data. And so what's important to note is that when you want to plot waveforms, you just have to say what channel it is that you want to plot. And then you have to tell EEGLAB what trials you want to plot. And to do that, you can use indexing. Once you've done that, you can compute the ERP averages. So you're just taking the channel that you want, the trials that you want, and you average over trials, and you can use this to plot. When you're interested in plotting a scalp map, it's a bit different. So rather than specifying the channel you want, you have to specify the time range that you're interested in. You also have to tell it what trials you want to extract, and then you compute the plot by averaging over time and trials, and you can use this topoplot function to plot it. So I'll cover this in more detail in a future video, but I'll just show you what it looks like. Cool. So this is the plot of that one participant that we've computed, and so this is the waveform at FCZ, time locked to the onset of the stimulus. The black trace is the standard trial, so that would be an even number, and the red trace is the odd number. So what normally happens in this task is you get an enhanced negativity to target stimuli, and that is evident here. And what I've plotted below is the scalp distribution of data between 250 to 450. So that's kind of capturing this enhanced negativity. And what you can see here is that there is a fronto-central distribution, and it's more prominent in the target trials, which corresponds to what the waveform is showing. And yeah, so that's how you would pre-process the data in EEGLAB using the graphic user interface. And in future videos, I'll cover how to pre-process the data using MATLAB script and how to plot waveforms and scalp maps in more detail. So yeah, thanks for watching, and I'll catch you in the next one.